school and workshop. I know how much work that is. So I've been working on this topic of population dynamics and evolution for many years now. And in the philosophy of the workshop, I decided not to focus on a specific paper or topic, but to give a general overview of the problems and types of tools that we use to do this, to try to understand these questions. So when we see origin of species, first thing that comes to mind is Charles Darwin, right? And his evolution by natural selection. So I know you are all familiar with this, but I would like to review this process just to stress three main ingredients that are always present in this type of process. So here I'm illustrating the evolution of the whales from a land mammal to a sea mammal. And so how does that work? So if we could go back in time and look at these populations, any population of these animals, first thing that we would see is that the individuals are not identical. So there is diversity. Individuals are different. So that's the first ingredient, diversity. And because they are not different, the ones that are better adapted to the environment, they grow stronger, they have more offspring, and they pass their genes to the next generation. So that's selection. And the third ingredient is mutations. So selection filters out part of the genome, part of the DNA, but mutations add novelty, add diversity, so that selection can keep working from generation to generation. And if you keep doing this for millions of years, you can have these amazing transformations from this animal that looks like a pig to a dolphin or a whale. But as you know, there is more to evolution than just this type of transformation. Here I'm illustrating this with a very simplified tree of our own species. And if you look here, you see that a group of primates evolved just like the whales into the humans. But a different group of the same population somehow took a different path and ended up as the bonobos. So at a certain point in time, there was this branching. And this is the speciation. This is what we're trying to understand. Under what conditions, what are the mechanisms that make this evolution? Because you may think that if the population is under selection, why they don't all follow the same path? Why it branches? So what can cause that? So that's the purpose of this general area of study. And so what the questions we want to answer is, what are the drivers of speciation? So first one is geographic barriers. That's one of the most important drivers. So suppose I have a population, and somehow an earthquake happens, and the earth opens, and a river appears and divides the population into two subpopulations. So you might think that on one side of the river, it's drier, on the other side it's more humid, the conditions are slightly different, the species evolve completely independent, there is no crossing between individuals of the two populations. We say that there is no gene flow between the populations. So evolution is independent, and after millions of years, it is inevitable that they will become different species. Even if the adaptation is slow, the random mutations, the mutations that happen here will be different from the mutations that happen there. They won't be transmitted. And if the river dries and they get together again, they won't recognize themselves as same species. So that's, we say that this is geographic isolation. It stops gene flow and leads to reproductive isolation. So reproductive isolation is my concept of species. So species are defined. This is a very controversial topic, how you define species, but for us, species are groups of individuals that cannot mate. Okay? And this process is called allopatry. It's patria, allopatria, different lands, they are geographically isolated. Nice example of this, is the finches that Darwin studied at the Galapagos Islands. So these birds, they migrated from the continent. They established themselves in different islands. They adapted. They could fly. There is always some level of migration, but they are essentially isolated in each of these environments, and they become these different birds. And here is what we call a phylogenetic tree of these birds. So they all came from a single common ancestor from the continent, and they branched like this. And one thing that I want to stress here is that there is a bar here which says genetic distance. And that means that different species are genetically different from each other. So these differences are encoded in the genome. And if you compare two individuals of the same species, there will be some difference. But if you compare individuals of different species, this distance is much larger. So genetic distance is a good measure of species, how species differ. But there are other examples of speciation where these geographic barriers are not so evident. For instance, in lakes, like this Lake Victoria in Africa, this is a big lake. And there are about 400 types of cyclids that live there. And these cyclids have the same common ancestor. And the origin of these species is, everything is controversial, but apparently they speciated in the lake. And in the lake, these barriers are not so evident, although the lake is very large, right? There are speciation in small lakes, also in Central America. So barriers may not be the only way to get that split. And there are also examples where speciation happens without space at all. They happen at the same spot. You have things happening in the same area, and two species emerge with complete gene flow. There is nothing forbidding people to mate individuals to mate in that area. And this is driven essentially by competition for food, for resources. There is a very nice paper that works out the theory and shows how this branching can occur only by competition for resources. So what we did, so we were interested in understanding how these things work in more detail. So we proposed a model that's a very simple model. I'm going to show it to you, how it works, what are the main ingredients, and what we get out of this model. And then later, I will show you applications that we did, more interesting applications that we did with this basic model. So the model is the following. Let me go right to it. Oops, here it is. So it's an individual-based model. So individuals are represented one by one. So each of these red squares is an individual. They are scattered in space. Space now, in this initial model, is a square with periodic boundary conditions. And to each of these individuals, we associate a genome, a DNA. So this is a very simple representation of the DNA. It's just a string with bits that are zeros and ones. You can think of this as a sequence of genes, and genes have two alleles. For instance, the gene for blood type has 3 alleles, right? A, B, O. Here, they only have 2. Every gene has 2 alleles, and it can be 0 and 1. And this sequence of zeros and ones, it encodes the genetic information for each individual. There are B of them. This is the number of genes. And we want to simulate the evolution of this population under sexual reproduction, recombination, and mutation. And this is how it works. So when an individual is going to reproduce, this guy here is going to reproduce, it cannot choose a mate anywhere. It's going to be local mating. So there is an area around the individual, a circle of radius S, and he's going to be able to mate only with individuals within this circle. S is a parameter. It can be very small, it can be very large. We're going to vary it to see what's the role of this local mating in the dynamics. But also, individuals can mate only if they are sufficiently similar genetically. So we are going to take the genome of this individual, we're going to compare it with the others in the neighborhood, and we are going to count the number of differences. If it's too large, if the genetic distance is too big, they cannot mate. So there's going to be a threshold G. So S is for space, G is for genetic. So there is a maximum genetic difference that allows them to mate. So the way it works is that it looks around, find the compatible individuals genetically, and choose one at random. Yeah, I'll get to that, yeah. So when they mate, what happens? You take the genome of the parents, and you do recombination. So for each gene, you pick the allele of the father or the mother. There is no sex, but they're all hermaphrodites for now. But you take the allele of the father or the mother with equal probability. You construct the genome of the offspring. And then on top of that, you allow each of these genes to flip with a certain probability rate, mu. Okay, so these are the parameters of the model. There is the mating neighborhood, the genetic threshold, the mutation rate, and the genome length. Actually, there are much more parameters. There's the number of individuals, the size of the area, lots of parameters, but these are the more important ones. And so we are going to keep the population constant, so at every generation. So generations are not overlapping. So we are going to do, every individual will have a chance to mate. When it mates, it produces an individual of the next generation. And there is also going to be a probability that the individual dies without mating. I'm going to call that probability Q. And when that happens, because we want to keep the number of individuals constant, we're going to pick a random individual in this area. It's like he died, there is food available, so we take another individual around here, and this guy has an offspring in place of this one. So this other individual can have two offspring when it's time to reproduce and when it replaces the other one that died. So not everyone has one offspring. Yeah. So this is a chance of not dying without reproducing. Initial condition is all individuals identical. And one last comment is that if you take this mating neighborhood to be the size of the system, and you let B go into infinity, the size of the genome, you go to this other model. It's called the Derrida-Higgs model, and Sabrina is going to use it. This is a nice model. It simplifies several things. You can get some analytical results that you can't otherwise. So, okay, so when you do that, this is what happens. So you start, you choose your parameters conveniently, and you start running this process, and you see the individuals that were identical start to get different, and with time, you see that species arise. So these different colors, they are species in the following sense. For instance, if you take the blue species and the yellow species, no blue individual can mate with no yellow individual. So the genetic distance between any pair, blue-yellow, is larger than your threshold G. So they are completely reproductively isolated, and you can call them species. So what's happening? Why is this happening? The idea is that, you see, there are no geographic barriers, but you have space, and the mating neighborhood is small. So if you have a mutation that happens here, this mutation can only be transmitted to an individual in the next generation, to an individual nearby, right? He's going to mate, and yeah, I forgot to say, when you create an individual, you place this individual within that radius S, right? So, so the mutation can go to another individual here, and then there. It's a diffusive process. It goes from individual to individual, but slowly. And so something that's happening, a mutation that's happening here, doesn't know about mutations that are happening here on the other side. And these mutations take a long time to talk to each other. So it is as if there were a barrier. So if the conditions are right, if the mutation rate is high enough, if this diffusion is sufficiently slow, that's what you get. About diffusion motion of the individuals, they don't move in a generation, but the offspring is placed within this area S. So it's like the family of that individual is moving in space. Then you can do several things. You can count how the number of species vary in time. So there is a time where number of species increases, then it reaches a plateau. There are always speciations and extinctions because I can get back to this later. And you can also change the parameters to see what happens. So these are snapshots of the system after many generations for different values of this mating neighborhood S and genetic threshold G. So if S is too large or G is too large, you don't get anything. So there's too much mix. So these mutations, they are traveling too fast in space, and they mix, and you don't get this split. So you need these restrictions to be sufficiently large so that speciation can happen. So this is, for instance, what is probably happening in these lakes, right? It's a big lake. There are no barriers, but you have this diffusive behavior of the mutations. And then you can compare this with numerical, with data, with empirical data. For instance, you do a simulation, and you see that there are some species with few individuals. There are species with lots of individuals. You can make a histogram of that. You do that many times. And you can calculate what is the number of species that has so many individuals. So typically, you have this number of individuals per species, but you have these tails. This you can compare with data. Here we are comparing with trees in this island. Of course, you cannot do this comparing ants and elephants, right? They have to be related species. They are species of the same clade. And other things. So you can do that. So this is an old paper. It's from 2009. It's our original model. And so what I've shown you, just check the time. Yeah. I've showed you speciation in space. I want to show you speciation in genetic space now. So what happens in genetic space? Genetic space is very complicated because it's the genome is a sequence of zeros and ones. So it's like the edges of a hypercube. And for 100 genes, you have 2 to the 100 possible sites, 10 to the 30. It's huge space. But there is a way to project this space in 2 dimensions. And you can do it for the best. You can find the best 2-dimensional plane in order to project that. So I'm going to show you these things. I'm not going to in detail how you can do that. But it's like, for instance, if you want to project a cloud of points that looks like the cell phone, you are not going to project it like this. You won't see anything. You have to project it like this. So there is a way to choose this axis. And here is a case where we have 3 species appearing. So at time 200, you have this cloud of points. So each point is an individual, and these axes are this strange projection of the genetic space. As time evolves, you see that the clusters, they go apart. I'm still painting them the same color because there is probably individuals here that I can still reproduce, so they're not completely isolated yet. Then now they split completely. I keep going. Now there is a 3rd species, and this dances in the genetic space. Remember that this is a projection, right? And now what I'm going to do is that I'm going to take all these frames and put them one on top of each other to have the time axis in the z direction. And this is what you get. So each cut here is one of the plots that you saw. And this is the tree, right? This is Darwin's tree. And the tree is thick because there is diversity, right? It's not thin. There is always diversity. And of course, the branches are overlapping because it's a projection. It's a nice way to view this process. So I know I have little time, but I'm going to go very quickly over three applications. One is ring species. This is very interesting. This is a paper from 2005, and it displays the a species of birds that's called the greenish warblers in Asia. So this is a big area in Asia, and the story is that these birds, they, like 10,000 years ago, they lived only on a forest on the south of the Tibetan plateau, because all the rest was frozen by the last glaciation. When the ice started to melt, these birds, the forest started to grow, and the birds followed the growth of the forest. But the forest could not get in here because this is too dry, so the forest grew into a ring. And the birds also moved around the ring, and after 2,000 years, as the birds moved, they changed their songs. And when these two fronts of birds met in the north, they couldn't recognize themselves as the same species. The songs were so different that they wouldn't mate with each other. But these guys here could mate with these ones with these ones, these ones, and so on. This is a recent break. Suppose that this is still connected. So you have this ring of individuals that can mate with each other along the ring, except here. Here there is this clash. So this is called a ring species. So we simulated that. So we changed the model. Number of individuals not constant anymore. Boundary conditions are not periodic. You have walls here. You have the plateau here. We added this extra barrier here to simulate these large areas in the north. And so you do this thing. And if you adjust your parameters right, you really get this formation of these ring species. And what we found is that, so we compared this with data. So this is data. This is our simulations. And what we found is that ring species, they form only for very fine tuning of the parameters. If the expansion time is too small, you don't have time to accumulate mutations. So the species, the birds are still the same, and when they get there, they are one species. If the expansion is too slow, it breaks into a species instead of generating this continuum. So ring species are very rare phenomena, and you can more or less understand it with this type of simulations. The other thing that you can do is to look at phylogenetic trees. For instance, when we do our simulations, we know when a speciation event happened, so we can mark it here. It's still growing. A third species appeared. Then it went extinct. So we can follow everything in the simulation, and at the end, we can build the tree. Let me skip this. And these trees can be quantified. You can have phylogenetic trees that are more balanced, or trees that are unbalanced, and there are numbers to measure this. This is an index that is used to measure balance. The larger the index, the more unbalanced the tree is. And you can also measure the distribution of length of these trees. So there are parameters that you can measure that tells you how these branches are distributed. And so what you can do is that we run our simulations. We have the full phylogeny. We can look at the species that survived at the last generation, and we can do all these measures. But what's important is that you can now change the parameters of the model and see how they affect the phylogeny. Because this is nice because you can maybe look at a real phylogeny and infer what is the type of process that gave rise to that phylogeny. So we did that. I'm going to just show you this final picture. So this is a picture where we have this balance index here, from unbalanced to balanced, from TP speciations at the recent times or at the beginning. And we do simulations varying the parameters, and you get these regions. So everything is stochastic, so every time you run, you get a different point. So these are regions varying the genome size and the mating neighborhood, and then you can place here empirical points. For instance, Darwin's finches are number 6. They're here. 9 is cyclids in one of these African lakes, etc. And then you can try to interpret what you are seeing according to the model. And last thing, I would just mention this and I'll finish. We also did, so you see, everything I showed you so far is neutral. There is no selection. There is only sexual selection. It chooses a mate that is similar to itself, but there is no real selection. Here is a case where we studied the interaction between the genome, the DNA that's in the nucleus of our cells, and the DNA that's in the mitochondria. You see, the mitochondria is very important because it is responsible for the energy of the cell. And in the process of doing this transformation from ADP to ATP, this process is very complicated. It involves proteins that are produced by the mitochondria. The mitochondria has a DNA, but also proteins that are generated by the nucleus. So there is an interaction between these two pieces of DNA, and they have to be sort of in equilibrium in order for the respiration process to work well. So we did this model. Now we separated males from females. We added mitochondrial DNA that is transferred only from the female to the offspring, and the nuclear DNA is recombined. And we measured how matched these two pieces of DNA are. If they are, for instance, we say that when they have the 0, 0, 1, and 1, they are okay. If you have a 1 and a 0 here, they are not okay. They are not working in synchrony. So we measure the mitonuclear distance and associate a fitness with that. So if the mitonuclear distance is 0, you have high fitness. If you have lots of divergence, you have low fitness. And this is interesting because you see, you can have a person whose nuclear DNA is very well adapted to its mitochondria. But then this guy is going to marry and have an offspring. His nuclear DNA is going to mix with his wife. And this mix might interfere with the mitochondrial DNA. Mitochondrial DNA is passed from the mother directly. So you might introduce incompatibilities and lower the fitness of the individual. So this is also related to speciation. So we made a study of that, see how this affects speciation, how it affects the balance of the trees, and so forth. We compared that with some data that we found. And there are very, very nice things. This is our latest paper that just came out, where we studied this, how selection works in these two DNAs. I don't have time to go into the details, but just to give you an idea of the type of things that we have been doing. So I think my time is up. I'm going to stop here, and if you have questions, I'll be happy to answer.
Thank you very much, Marcus, for this wonderful talk. So we have time for questions.
Thank you, Marcos. I love it, like always.
Thank you.
Big fan. I have two questions. The first one is whether this preference of mating with someone similar is also genetically coded, or whether you could include the lack of that preference. And the second one is in the 2005 paper with the warblers, where when they met, they were still genetically compatible and they just did not meet
That's
because
right.
of the
That's
song.
Right. Yeah, right. Yeah, good. Both good questions. Yeah. First one. Here we assume that this mating choice is already there. We don't simulate it, but you can. So there are papers where they use similar models, and they show that this can emerge naturally. You have another piece of DNA that when it's all ones, you have mating preference. When it's all zeros, you don't, and it evolves in that direction. That it can be done, but we don't do it here. About the other question, that's very interesting. It also happens with the fish in the African lake. They don't mate because they don't want to. But if you put them on a tank and turn off the lights, they won't see each other, and they will mate, and the offspring is viable. So it's the same thing. So this is, so the question is, this is always a question that I have for myself. What are these numbers here? Are they really genes? Are they letters in the DNA? For me, I take it as a representation of the genetic information. So you can say that it codes much more for the phenotype than for the genotype. So the birds, they have red feathers and blue feathers. They could mate, but they say, no, I don't like to mate with that one. But there are questions of interpretation that are not completely clear to me. Thank you. Yes. Press the button, yeah.
Okay. Thank you for the talk. Very interesting. So in your model, the individuals don't move, right?
Yeah. What happens is that they don't move at that, so they live there. But when they mate, the offspring is placed inside this neighborhood. So the offspring is going to move. So if you look at the points, generation after generation, they will be moving around. Yeah. But it's not the individual, it's the offspring that is. Yeah.
So I'm just wondering how close that is to, for example, the fish case in the lake. Do the fish really live their life in a
Yeah.
fairly small neighborhood?
Yeah,
And-
mostly they do. But, and also, this can also be more, for plants, for instance, this is more relevant. But even for fish, the fish, they
Yeah.
don't travel the lake. They, yeah, but,
And-
yeah.
I also had another question. I'm wondering about how the time scale of speciation depends on the length of the
Of the genome,
genome.
yeah.
And because like you at the beginning you said that there are 400 fish species with a common ancestor from 14,000 years ago, which seems
Yeah,
very
that's
recent.
debatable. There is a lot of papers about that. Some people say it's not 14,000, it's 1 million years.
Okay.
But it's still very recent. I think 4,000 years is ridiculous. I mean, it's true, yeah. But I don't know, but yeah.
Okay, so in the interest of time, let's think
Yeah.
again.